Hello, everyone. Today, I'm going to introduce GenStore, a high-performance and storage processing system for genome sequence analysis. Okay, so genome sequence analysis is critical for many applications, such as personalized medicine, outbreak tracing, and evolutionary studies. Genome sequencing machines extract the smaller fragments of the original DNA sequence, known as reads. So read mapping is the first key step in genome sequence analysis that aligns reads to potential matching locations within the reference genome. And for each matching location, the alignment step finds the degree of similarity between the read and the reference by calculating alignment score. So calculating alignment score requires computationally expensive approximate string matching, or ASM, to account for differences between the read and the reference genome that might occur due to sequencing errors and genetic variations. So read mapping performs alignment on large genomic data sets containing millions of reads. Therefore, read mapping is both computationally expensive and incurs high data movement overhead from storage. There's been significant effort into improving read mapping performance through efficient heuristics, hardware accelerators, and various filters that prune reads that do not require expensive computation. And while these approaches address the computation overhead in read mapping, none of them alleviate data movement overhead from storage, whose impact becomes even larger when the computation overhead gets alleviated. So our key idea is to filter reads that do not require expensive alignment computation in the storage system to fundamentally reduce data movement overhead of read mapping. Examples of reads that do not require costly alignment step are exactly matching reads to the reference genome that do not need approximate string matching performed during alignment, and non-matching reads that do not have any potential matching location in the reference genome and can skip the alignment step. However, filtering reads in a modern SSD can be challenging due to different behavior across read mapping workloads and limited resources available in the SSD. By addressing these challenges, we propose GenStore, the first in-storage processing system designed for genome sequence analysis to reduce both computation and data movement overhead. GenStore provides high performance and energy benefits compared to state-of-the-art hardware and software baselines. So that was a summary of my talk. Let's start with the background on read mapping so we can dive into the talk in more detail. Mapping reads to reference genome requires expensive computation on large data sets. The search space in the reference genome can be very large. For example, for a human reference genome, it contains more than 3 billion characters. Therefore, read mappers typically use an index of the reference genome to reduce the search space. The index contains unique K-length subsequences called k-mers extracted from the reference genome and the locations of these k-mers in the reference genome. So read mapping is a three-step process, and the state-of-the-art read mappers usually involve several heuristics to reduce the cost of expensive alignment computation. In the first step, called seeding, the mapper extracts k-mers from the read. So the goal is to determine potential matching locations or seeds in the reference genome. And to do that, the mapper extracts some k-mers from the read and looks up the k-mers in the index. And in case the k-mers hit in the index, the mapper marks the locations of those k-mers in the read as potential matching locations or seeds. So instead of looking at the whole reference, we can only look at these small seeds, these candidate locations. And in the second step, to further prune the potential locations to check or to prune some seeds, seed filtering or chaining gets performed to further prune some of these seeds. And it is done usually with a less computationally expensive approximation of alignment score. And finally, the third step is alignment to determine the exact differences between read and the reference genome for the reads that pass through the second stage. So if a read does not have any seeds remained after the second stage, it gets filtered. Otherwise, it goes to these third stage, which requires the expensive approximate string matching to find the exact differences. So we perform experimental studies to understand the potential of efficient in-storage filters for improving read mapping performance. And we perform a case study on real genomic read dataset and various read mapping systems, both hardware and software, and state-of-the-art SSD configurations. And based on our motivational analysis, we conclude that the ideal in storage filter significantly improves performance by both reducing computation overhead and reducing data movement overhead. Filtering outside SSD, on the other hand, provides lower performance benefits since it does not reduce data movement overhead and must compete with read mapping for other resources in the system, such as mid-memory bandwidth or computation resources. So a hardware accelerator mapper reduces the computation bottleneck, which in turn makes I/O a larger bottleneck in the system, and therefore makes the stronger case for filtering reads inside the SSD possible. So motivated by these observations, our goal in this design, in this work, is to design a storage filter for genome sequence analysis in a cost-effective manner. And we have 3 key design objectives in mind. First is that the system should provide high in-storage filtering performance to overlap the filtering that is happening in the SSD with the read mapping that is happening either in the software system or like in a software-based read mapper in a CPU or in an accelerator. So wherever the mapper is happening, the filtering, the goal is to overlap and hide the latency inside the SSD. So, and the second goal is that the design should support reads with different properties and different degrees of genetic variation. And third, it should require significantly, it should not require significantly high hardware overhead. So low costness is the third goal. And to this end, we propose GenStore, which is the first in-storage processing system designed for genome sequence analysis. And the key idea is to filter reads that do not require alignment inside the storage system and send the unfiltered data to the host system for further processing. However, filtering reads in a modern SSD, as we discussed, can be challenging due to different behavior across read mapping workloads and the limited hardware resources available in the SSD. So let's take a look at filtering opportunities based on the input reads. So sequencing machines typically produce one of 2 kinds of reads: short reads that are highly accurate but short, for example, up to a few hundreds of DNA characters, and long reads that are less accurate and long, from hundreds to millions of DNA characters. Based on these, we leverage 2 filtering opportunities. First, we can filter exactly matching reads, which are reads that match exactly to one or more subsequences of the reference genome, and therefore do not require approximate string matching during alignment. And based on simple computations, basically can be filtered inside the SSD. And examples of these exactly matching reads can frequently occur in short read sets with low sequencing errors and also low genetic variation. Second, we can filter non-matching reads, which are reads that do not have any potential matching locations in the reference genome and can skip the alignment step because there is, from the second step of read mapping, they already don't have any location to go through the third. So they can also be filtered. So such reads, non-matching reads, can frequently occur in long read sets with very high sequencing errors, and short or long read sets that have high degree of genetic variation and basically would align to no place in the reference genome. So by thorough analysis of mapping processor reads with different degrees of genetic variation and different properties, we designed 2 low-cost storage filters: GenStore-EM for filtering exactly matching reads and GenStore-NM for filtering the non-matching reads. So let's take a closer look at GenStore-EM. GenStore-EM accelerates read mapping by using an efficient in-storage filter to filter reads that have at least one exactly matching location in the reference genome, via simple operations without requiring expensive alignment. So the key challenge, however, in designing GenStore-EM is the large number of random accesses to large data structures inside the SSD. And this is challenging because NAND flash memory exhibits poor performance for random access reads. And there's limited DRAM capacity available in the SSD, which is relatively small compared to the size of data structures that need to be accessed. So to reduce the number of accesses per each read, we introduced the concept of read-sized k-mers. Therefore, instead of having separate k-mers extracted from the read, we can consider the whole read as one k-mer. So we can have a read length k-mer, so that we can have for each read only one index lookup per read. So now that we only have one access per read, we can avoid also random accesses to the index by introducing sorted index of read size k-mers. So this sorted index allows finding exact matches via sequential scanning of the read sets and the index. So I showed the key idea of GenStore-EM with a simplified example in which each short read consists of 10 characters, but in reality, that can be a few hundred characters. So here we have this 10-mer that is like one read. So suppose we have two data structures. One is a sorted read table, each entry of which stores a read and its unique ID, and a sorted k-mer index, which contains all unique read-sized k-mers extracted from the reference genome, along again with the location of the k-mers in the reference genome. And each data structure is sorted by read and k-mer in alphabetical order. So we sequentially scan through these data structures in 3 different ways based on the comparison result of the current read and k-mer. So first, when the current read and k-mer are identical, we record the read as an exactly matching read that can be filtered from further read mapping process, and then we move to the next element in both arrays. And if the read is alphabetically larger than the k-mer, we conclude that a k-mer does not match any read and go to the next element in the index so we can examine the next k-mer. And finally, if the k-mer is alphabetically larger than the read, we conclude that the read does not match any k-mer in the index and need to be sent to the full read mapping process because we've been sequentially scanning. So if there was any match, we would have found it by now. So, okay, that's a non-matching read. So then we go to the next element in the read data structure so that we can check the next one. So using this technique, GenStore-EM avoids random accesses and performs filtering using only simple low-cost logic. Despite the key benefits of the sorted resize k-mer index, the index actually takes up a larger space. For example, for humans, it can take up to 126 gigabytes to index it with such large k-mers due to the large number of unique read-sized k-mers. So we further reduce the overhead of GenStore-EM by replacing read-sized k-mers with a strong hash of each read that can act as sorting criterion and the fingerprint of each entry. So using strong hash values instead of read-sized k-mers, we can reduce the size of the index by 3.9 times. While still this index is larger than a baseline k-mer index used in conventional read mappers, our proposal is feasible for in-storage processing due to large capacity and high internal bandwidth of modern NAND flash-based SSDs. So basically, that is a solution tailored for this architecture. So now I show the overall operational flow of GenStore-EM with sorted read table and sorted k-mer index in the NAND flash memory distributed across channels and dies. And since they're evenly distributed across channels, dies, and planes, we can leverage the full internal bandwidth of the SSD. And then we have also the comparator logic located on the SSD controller. So GenStore-EM consists of 2 steps. Step 1 reads the 2 data structures from NAND flash chips to the SSD's internal DRAM in a batched manner. And step 2 performs exact match filtering within each read batch using only simple comparator logic. Step 1 and 2 are performed in pipelines manner, and during filtering, since this is working in a pipeline manner for different batches, GenStore-EM sends the unfiltered reads to the host system to do the full read mapping whenever it detects like unfiltered reads for the batch. And then meanwhile, for the next batch, GenStore can filter the reads again. So it can fully work in the pipeline manner. So now let's take a closer look at GenStore-NM. So using chaining, chaining or seed filtering, which was the second step of read mapping that we discussed, it filters most of the non-matching reads, which are reads that would not align to any subsequences in the reference genome. And recall that chaining filter calculates a similarity score for each read, which is called chaining score, and it filters reads with no high scoring potential matching location. So calculating a chaining score in SSD is challenging because finding the best chaining score requires performing many iterations of dynamic programming, a dynamic programming algorithm for all seeds within a read. And this can actually particularly be challenging for long reads since they can have a very large number of k-mers, like up to a few millions. So to reduce the cost of chaining, GenStore-NM uses a lightweight chaining filters that selectively performs chaining only on reads with a small number of seeds and directly sends reads that require more complex chaining to the host system. This idea is based on our observation from analyzing a wide range of real-world genomic data sets. So this figure here shows alignment probability of a read in a long read set to subsequences in a reference genome as a function of number of k-mers per read. So we observed that reads with sufficiently large number of seeds are very likely to align to subsequences in the reference genome. And such reads can directly be sent to CPU or like an accelerator for full read mapping and bypassing the in-storage filter, because anyways, with high probability, they will not be filtered. So we observed that reads with sufficiently large numbers, so they can easily be already bypassing. And then we conclude that the selective lightweight chaining approach can filter many non-aligning reads without costly hardware resources in the SSC. And for more details about it, please refer to our paper. Now I go to our results. So we evaluate the following systems. BASE is the state-of-the-art software or hardware read mappers for both short and long reads, and GenStore, which is BASE integrated with GenStore inside the SSD. And we evaluate these mappers and systems with various SSD configurations, a low end, a medium end, and a high end SSD. And for other details about our methodology, again, please refer to the paper. We analyzed the benefits of GenStore-EM for a 22 GB short read set where 80% of reads exactly match some subsequences in the reference genome and can be filtered. So that typically happens in let's say, a human data set. So we show the benefits of GenStore on hardware and software mappers. So GenStore provides up to 2.5 times the speed up compared to software baseline and up to 3.3 times speed up compared to the hardware baseline. And it provides an average 3.9 times energy reduction. And we also analyzed the benefits of GenStore-NM for a 12 GB long read set with very high degree of genetic variation compared to the reference genome, where 99.7% of reads do not match any subsequences in the reference. And we show the benefits of GenStore on software and hardware read mappers, and show that it provides up to 27.9 times the speed up compared to the software baseline and up to 19.2 times the speed up compared to the hardware baseline, and on average 27.2 times energy reduction. So we find the area and power values of GenStore by synthesizing GenStore-EM and NM using 65 nanometer technology node, and find that for an 8-channel SSD, the area of GenStore is 0.2 millimeters square, and power is 26.6 milliwatts, sorry. So by scaling the area to lower technology nodes, we observed that the area overhead of GenStore is 0.006% of an Intel processor and less than 9.5% of only the 3 ARM cores that are present in a SSD controller. So let me briefly talk about the results that are included in the paper before like other results that are included in the paper before I conclude. So these results include the following: effect of read dataset features and performance. For example, the data size, which we scale up to 440 gigabytes, the filter ratio, basically how many % of the reads in a read dataset can be filtered, performance benefits of an implementation of GenStore outside SSD. In some cases, we observed that it actually provides benefits because of its more efficient streaming accesses. And also, right, so because of that, it provides benefits for some cases, but it significantly provides lower benefit compared to GenStore implementation inside the SSD. And also, we provide more detailed characterization of non-matching reads, basically when they happen and what are the scenarios that they happen and by what degree of, I don't know, filtering is possible across different read mapping use cases and also species. So now let me conclude by giving the summary of this talk. So there's been significant effort into improving read mapping performance through efficient heuristics, hardware accelerators, and various filters that promote reads that do not require expensive computation. And while these approaches address the computation overhead in read mapping, none of them alleviate data movement overhead from storage, whose impact becomes even larger when the computation overhead gets alleviated. And our goal is to improve the performance of genome sequence analysis by efficiently reducing unnecessary data movement from the storage system. And the key idea is to filter reads that do not require expensive ASM computation in the storage system to fundamentally reduce data movement overhead. However, filtering reads inside the SSD is challenging because read mapping workloads, as we saw, can exhibit different behavior for different types of read data sets, and there are limited hardware, available hardware resources in the storage system, such as the limited DRAM capacity that we talked about. So to this end, we proposed GenStore, the first storage processing system designed for genome sequence analysis to reduce both computation and data movement overhead. And GenStore provides high performance and energy benefits compared to state-of-the-art hardware and software baselines. So that concludes my talk. Thank you very much for listening, and I can take any questions if there's any.
Thank you very much, Nika. Are there any questions in the room? Okay, I just want to ask a very quick question, Nika. So you focus on genome sequence analysis. Are there any other stages of the genomic pipeline or bioinformatics pipeline that you think could be a good match for in storage processing?
Right, yeah, that's a very good question. So another step that we're interested into looking at are steps before even read mapping, because in some cases, you might even not know what reference genome you should pick to align your sample to. So you need to first do some metagenomics classification to figure out basically from pool of very large set of references, select some relevant references so you actually can do read mapping on. So which is, as I said, refers to as like metagenomics classification, which is again a very important step. And it's we predict that would also be very beneficial to look into those steps to even, because that even deals with larger scale data. So we're excited to look into that too.
Okay, that makes sense. Thank you very much, Nika.
Sure, thanks, Juan.